7th period and my 8th period have the same explanation that you do so that it's fair. So, the first step in cell respiration is called what? Glycolysis. What molecule do we start with in glycolysis? Glucose, which has 6 carbons. So 1, 2, 3, 4, 5, 6. Is that 7?
That's 7.
Just kidding.
You're a bad math teacher.
That's why I'm a science teacher.
And you get it recorded.
Great. Now 4th and 8th grade, we'll get to see that too. What are we going to do with glucose in glycolysis? We're going to break it in half, and it becomes what?
Pyruvate.
Pyruvate, which is how many carbons? 3 carbons.
No.
1, 2, 3. Okay, so in the process of breaking down glucose into 2 pyruvates, what energy do we get out of this?
4 ATP.
We get 4 ATP. Did we use any ATP?
Yes, we do.
Okay, so we made 4 ATP. We used... Okay, so everything you're doing that's annoying right now, on video. Just so you know.
Oh.
Yeah, thanks. Okay, ATP used was how much?
2.
2. Okay, do we use ATP or any type of energy in any of the other processes in cell respiration?
Nope.
No, this is the only time you will actually use energy. All the rest of it, we're going to gain. Okay, what else did we make?
NADH.
NADH. How many NADHs did we make in glycolysis? We made 2. Okay, is that going to turn into ATP right now?
No.
No, we have to take it to what step? Electron transport chain, which is part of oxidative phosphorylation. Okay, we don't turn it into ATP right now. That's what people are kind of struggling with right now. Right now it's just NADH. It's just full electron carriers. Okay, it's not usable ATP yet. Okay, so we'll take that. We're going to bank that, and we're going to keep that for later. What do we do next with pyruvate?
Break it into acetyl-CoA.
Break it into acetyl-CoA. How do we do that?
You take off 1 carbon.
Take off a carbon. Where does that carbon go?
Outside the respiratory.
It goes as waste? In what form?
CO2.
CO2. Okay. Do I do it to this one too?
Yes.
Yep. Everything we do, we're doing twice right now. So that leaves us CO2. Okay. Now I'm left with a 2-carbon molecule. Well, I'm left with 2 2-carbon molecules. What am I going to do with these 2-carbon molecules?
Put it through the Krebs cycle.
What do I have to add to it first, though? Okay, so these 2 carbons right here, this is where we get the name acetyl. So the reason it's called acetyl-CoA is because these 2 carbons, which are called acetyl, join with coenzyme A, which we just abbreviate as CoA. Okay, so CoA by itself is just called coenzyme A. Okay, these 2 carbons are why we call it acetyl. A-C-E-T-Y-L, acetyl-CoA.
I thought it had an E.
Nope. There's an E right there.
Come on, the end after L.
Nope. Okay. In the process of turning pyruvate into acetyl-CoA, first of all, what's this step called?
Krebs cycle.
Linking stage.
Linking stage, link reaction, any of those are fine. Okay. What did we get out of this? What kind of energy did we get out of this?
2 NADH.
2 NADHs. Did we get any ATP? No. So does that mean this NADH is going to be ATP yet?
Later.
No. Later. Again, that one's going to be banked for later also. Okay. How do we feel about glycolysis and the link reaction? Good? Okay, now it's time to ask questions if you have them. Yes? Oh, okay. Okay, next step is called what?
Krebs cycle.
Krebs cycle, or what else?
Citric acid cycle.
Or citric acid cycle. Okay? What do we start the citric acid cycle with? What molecule do we start with? Acetyl-CoA, which has 2 carbons and a CoA group. Okay? How many of these do we have? 2. So remember that everything we're doing now is still happening twice. What are we going to do with this acetyl-CoA? What's going to happen to it as it goes through the citric acid cycle?
That's an amazing circle.
Carbons.
Huh?
They'll break and combine with carbons.
Okay, so what are we doing with carbons in this system then?
Getting
we're going to break it, but what do we, so there's three things. Remember, there were three things that I said happen within the citric acid cycle. One starts with an R.
Oh, yeah.
Yeah. Rearrange. So we're going to rearrange carbons.
Oh,
So we're going to move them all around. We're also going to do, what else with carbons? We're going to start with the letter A. Okay, if we attach them, where do we get them from? Add. What? What? Yeah. Add carbons. What else are we going to do with carbons? Subtract carbons.
I like that.
I like math too, especially when it's add and subtract. Okay? So, within this cycle, okay, right here we add, I think we add like 4 carbons. I'm not making you memorize that part, but a lot of people are also really struggling. People are thinking that the reason we're getting CO2 out of here is because, oh, this acetyl-CoA is going to come in and it's going to break apart and they're going to leave this CO2. That's not how it works. Okay? These 2 carbons are going to add to more carbons, then they're going to rearrange, and then we're going to lose a carbon in the process, and then as we keep going, we're going to lose another carbon in the process. Okay? Remember, all the carbons leave is carbon dioxide. Does that make sense? Huh?
Right there,
Yeah, we added 4 carbons to this 2 carbons, so we have 6. And then we go down to 5, and that's why we lose the CO2 here. And then we go down to 4 carbon molecule right here, and that's why we lose the CO2. So that's part that I'm not having you memorize, but you also need to understand where these CO2s are coming from. They're not just coming from acetyl-CoA. Okay? So within this, we are also making energy. How much ATP did we make?
2.
Is that 2 total or 2 per cycle?
2 per cycle.
2 total, remember? We made an ATP. We made 1 ATP in a cycle. Then we did the cycle again, so we get 2 ATP. Okay? We also made an ADH.
If it was 4, if it was 2 per cycle, Wilson, then it would be 4.
Yeah, it would be 4 total. How many NADH did we make?
6.
6 total or 6 per cycle?
So,
6
total.
total. Okay?
Otherwise
So in
it'd be
one
12.
cycle, we have 1 NADH made.
2,
We have 2 NADHs made.
2,
And we have 3 NADHs
3.
made. Anytime you have an arrow, you've seen arrows a lot, especially dealing with cell respiration. The arrow going in is the thing you use. The arrow coming out is the thing you get out of it. Okay? So in this case, we're getting out carbon dioxide. We're getting out NADH. We're getting out ATP. What's the last thing that we get out of the citric acid cycle?
FAD.
money.
FADH what? 2. Don't forget that it has a 2 at the end.
Everything has 2 citric acid cycle.
How many FADH2s do we have? 2. Per cycle or total? Total. Okay, so we also had an FADH2 made over here. Okay? Is this usable ATP yet? Nope. Now where do we go to make it usable ATP? Oxidative.
Phosphorylation. This is a long video.
Phosphorylation. That's alright. It's a lot of good explanation.
Special.
Yep. So we have all these full electron carriers, right? We have our NADH, and we have our
It's a butt.
FADH2. Thanks, Hazel. That's on video.
I know.
And these are full of electrons, which are still attached to hydrogens, right? Anytime I'm talking about electrons, I'm also talking about hydrogens.
Yes.
Okay, so now we're getting to something called,
Water.
you tell me what's it called.
Water
What's
with
the first
triangles.
part?
Electron transfer chain.
Electron.
Transfer chain.
Chain.
Transport. Chain. Not train. Chain.
Who said train?
I've heard chain or train random times. So now we are going to drop off these hydrogens that are attached to electrons so they can move through the electron transport chain. What are they going to do? First off, what does the electron transport chain do by its name? Transports what? Electrons.
No way.
Okay? What else is it going to do? Remember, we bring our electrons attached to hydrogens into here. We're going to separate it, yes, Andy, into proton and our electron, which is negatively charged. Huh?
No, that's
Okay? We had electron, our neutral electron right here that we were carrying through. Now as we bring it over here, our hydrogen with an electron attached to it, it's neutral. Now as you bring it through here, if we strip off that negative charge, you have to be left with a positive charge. Okay, because they have to balance each other out. Okay?
Why is
So,
one in H?
what?
Why is one in H?
What do you mean?
Why is one in H?
They're all H's.
And why is the other one a dot?
Because that's an electron. That's the electron that we stripped off of the hydrogen. Because anytime, like in the periodic table, you know how hydrogens are in the first column? Okay, when the hydrogens are in the first column, that means they have one electron attached to them. So this whole time, anytime I talk about electrons, I'm also talking about hydrogens.
So you said only the electrons were through.
Huh?
I thought you said only the electrons were
No, 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 no. As it goes through here, then it becomes electron, hydrogen. Anytime I'm talking about electrons, I'm still talking about hydrogens. So yes, that is confusing. You weren't here for the first one, that's why.
That was on film.
So yes, this is hydrogen attached to electron. When we get to here, hydrogen no longer attached to electron. Okay? What we're worried about now, we don't really care about the electrons anymore. We're done with them. Okay? What we're worried about now are all these positively charged hydrogens. We just call them protons now.
Yeah.
It's a single positively charged molecule. Okay? Once we have all those positively charged hydrogens, they're going to start building up over here. As they build up over here, they're going to start moving through this ATP synthase. Synthase.
Yeah, I know, that's what I said.
Yep. On the other side of that ATP synthase are a bunch of free-floating molecules
Holy Cosmos.
of ADP and phosphate. Remember, anytime we have the P, that means phosphate. Okay, these are all just free-floating around. They're just like, they're just kind of close to each other, but really they're like, stay away. Don't get too close. I need my personal space. ATP synthase, synthesis, make ATP. Okay, so when these hydrogens move through the ATP synthase, it's going to cause ADP and P to find each other. And then when ADP and P find each other, we make usable ATP. Stop playing your guitar. Okay? So from just the electrons, remember we got 34 ATPs. Okay, so all those NADHs, all those FADH2s we were banking, now we brought them over here, we cached them in. We made ATP with them. We made 34 ATP. But how many total ATP do we get from one molecule of glucose? 38. So how do we get that other 4 ATPs? Where do these come from?
Glycolysis.
How many come from glycolysis?
2.
2 from glycolysis.
Krebs.
Oops, I spelled that wrong. Y-S-I-S. 2 are from glycolysis. And 2 are from Krebs. Yeah?
Yeah.
Any questions? 34 all came from oxidative phosphorylation. Right? So we had 4 ATP in the beginning. That wasn't that much. So we had to wait till the very end to cash it all in and make it actually worth something. Any other questions? Okay, good. Stop.
Bye.